Hello class, this is Dr. Anderson and I'm putting a recording together prior to our endocrine class. I'm going to review basic endocrinology for you to refresh yourselves before class, and we will be spending class on aspects of endocrinology. So first of all, a hormone is defined as a chemical substance secreted into body fluids. Typically when people are thinking about endocrine substances, they're thinking about the blood, but hormones can be secreted into the extracellular fluid and talk to a neighboring cell. An endocrine gland is one cell or a group of cells that produces a hormone. So for example, there were one-celled glands in the wall of the GI tract that could produce endocrine secretions, or you could have a whole gland, the islets of the pancreas or the parathyroid gland, or any number of things. That chemical agent goes into body fluids, particularly the blood. It's going to travel to its target tissue, and what determines if it's a target tissue? The presence of a specific receptor. So I don't think of renin as a hormone because it's an enzyme. It's not working on a specific receptor. Alrighty, so we have synaptic transmission. We talked about it in our first semester. We have a signaling cell that's a neuron that releases a chemical agent across a synapse. So a chemical synapse is a one-way transmission. So something like dopamine could be released from a neuron and then act on a target cell. Here we have a paracrine agent. Paracrine agent, we find a lot of this in the immune system. You have a cell that produces a prostaglandin. It diffuses through the extracellular space and then works on a nearby cell through that endocrine agent. So nitric oxide could be a paracrine agent. Nitric oxide could be a neurotransmitter if it came from a neuron. So different molecules can wear different hats. Now our traditional view of a hormonal secretion or an endocrine gland, you have an endocrine gland that produces hormone that goes into the blood, travels some distance. The hormone diffuses out of the capillary system, finds its target, and the target is identified by a specific receptor. There are also cases, and we also find this in the immune system, where you have a cell that produces a substance, and that substance turns around and interacts with the very same cell. And we have a name for that, and that's called autocrine. Our discussions are going to center around the traditional definition of an endocrine substance. So many of our endocrine systems start in the hypothalamus. This is how the brain can interact with the endocrine system. So you can have stress or various stimuli having their effects on the hypothalamus. The hypothalamus then can produce a chemical agent that goes through a portal system and then interacts with the anterior pituitary. Anterior pituitary produces a number of protein hormones. So adrenocorticotropic hormone, or ACTH, which is then going to have its effects on the adrenal gland. Thyroid stimulating hormone, or TSH, which is then going to have its effect on the thyroid. Gonadotropin releasing hormone then works on from the hypothalamus, causes the release of hormones we call gonadotrophs, and there are two types of gonadotropes: follicle stimulating hormone and luteinizing hormone. These are produced in both males and females and work on both the testes and the ovaries. The interesting thing is that FSH and LH were isolated and purified first in female animals and given female names, and then the research in the males revealed that males have the very same peptide hormones. The Anterior pituitary produces a substance called prolactin that promotes milk secretion. We're going to find there's a hormone that we sometimes think of as a neurotransmitter, but in this case it's a hormone. Dopamine inhibits prolactin, and then that inhibition is removed so that prolactin can act on the breast. Anterior pituitary produces growth hormone, which then goes out to the liver, goes to the bones, goes to the muscle, and we get another hormone called IGF. All right, so we've talked about a number of peptide hormones that come from the anterior pituitary. In the hypothalamus, there are also neurons that project down to the posterior pituitary, and those neurons then release substances from the posterior pituitary. It's merely a release site, so the posterior pituitary is thought of as an embryonic extension of the hypothalamus, and there are two hormones that are released in that way. We have talked about one, ADH, or antidiuretic hormone, working on the collecting ducts to favor the reabsorption of water, and then oxytocin. Oxytocin has a number of functions: milk letdown and contraction of the uterus in labor and delivery. Oxytocin and ADH, interestingly enough, have central nervous system effects. ADH tends to increase anxiety. Oxytocin tends to decrease it. So a lot of work is being done to try to flesh out the actions of these peptide hormones on the central nervous system. If we talk about the mechanisms of hormones, we need to distinguish if we're talking about a lipid-soluble hormone or a water-soluble hormone. Lipid-soluble hormones don't travel in the blood very far on their own because they don't dissolve in blood very well. So they're carried around by a transport protein. So for example, testosterone is carried around by sex-binding globulin. Cortisol is carried around by cortisol-binding globulin. There's a thyroid-binding globulin. So these can be quite specific, or the transport protein could be albumin. Albumin is a very much an all-purpose carrier. The transport carrier gets the hormone to the target, but then we have to have a dissociation. The lipid-soluble hormone then can diffuse into the cell and find an intracellular receptor, and many times that receptor is actually in the nucleus. And what you see are two protein parts that come together as a dimer and then bind to the steroid. This is not an unusual way. So estrogen certainly works this way. Progesterone certainly works this way. The complex then finds a hormone-sensitive element on the DNA that promotes transcription of the DNA into messenger RNA. Messenger RNA then goes out into the cytoplasm and is then transcribed, whoops, I used the wrong word, and then is translated into a protein. That protein has to be folded and activated, but we get new protein synthesis from gene expression, usually from lipid-soluble molecules. So this is certainly how estrogen and progesterone and testosterone work. This is how cortisol works. This is how aldosterone works. There is some evidence that steroid hormones can have plasma membrane receptors. We don't know a lot about that. But typically we associate steroid hormones with gene expression and a process that takes quite a while to see the effects of. Water-soluble hormones don't need a carrier. They dissolve in the blood quite well. They leave a capillary system, find a specific receptor that's in the plasma membrane. Typically they activate a second messenger system, and many times that second messenger system is cyclic AMP. So glucagon works through cyclic AMP. Adrenocorticotropic hormone works through cyclic AMP. ADH works through cyclic AMP. Lots of hormones work through cyclic AMP. Glucagon works through cyclic AMP. We have some exceptions along the way. Atrionatriuretic peptide works through cyclic GMP. Dr. Olson taught you about insulin. Insulin works through a tyrosine kinase, but cyclic AMP is a very widespread second messenger system. And hopefully you remember how this works. You have a G protein that is activated through the binding of the ligand. That activates a hormone, or an enzyme, called adenylate cyclase or adenylyl cyclase. That catalyzes the reaction of ATP to cyclic AMP. So cyclic AMP is the second messenger, and it works with a protein kinase A to activate some proteins, inactivate other proteins. You could be moving organelles around inside the cell along the cytoskeleton. You could be encouraging exocytosis or calcium release or any number of things. Now, second messengers can also lead to transcription factors. So a hormone like growth hormone not only has short-term effects through a second messenger, but has longer-term effects through transcription factors. So there's quite a bit of crossover between a water-soluble peptide hormone and a lipid-soluble steroid hormone. If we sort of summarize steroids versus proteins, a steroid is going to work on an intracellular receptor, usually an intranuclear receptor even, promote gene transcription. It's carried around on a protein carrier, which slows its ability to be broken down. And therefore, steroids, because they work through gene transcription, are slow acting, and because they're shielded from degradation by their protein carrier, they tend to be longer lasting and have longer half-lives in, at least an hour and a half, and something like estrogen, its half-life is 13 hours, something like T3 has a half-life of 24 hours, so longer half-lives because they're shielded from degradation. Proteins, on the other hand, tend to work through extracellular receptors in the plasma membrane. They activate the production of an intracellular second messenger. They're carried freely in the blood. So because they're working on a second messenger and activating proteins that are already present, they work very rapidly, and because they're not shielded from degradation by a protein carrier, they are broken down quite quickly. That's a good thing. Insulin is broken down very rapidly because it's a peptide. That's a good thing. You produce insulin, you lower the blood glucose level, you want to break down the insulin and get rid of it. You don't want insulin to keep acting and set the person into hypoglycemia. There's a very good reason for this timing. Hormones interact, and a major way hormones interact is through an effect called permissiveness. So when a hormone is permissiveness, hormone A is permitting B to work. It doesn't do the job, but it must be present for hormone B to do the job. So insulin is permissive for growth hormone to cause growth. How does it do that? Well, growth hormone promotes protein synthesis. Insulin helps you get amino acids into a cell so growth hormone can work better. Growth hormone and cortisol are both permissive for glucagon. So growth hormone and cortisol are doing things that glucagon needs in order to raise the blood glucose level. Here's a diagram from Vander showing you permissiveness. So thyroid hormone all by itself doesn't do much in the way of lipolysis because we don't see a lot of fatty acids being released. This is not any different than the control. Epinephrine binds to beta receptors, causes some fat breakdown, so it clearly has an effect. But if thyroid hormone is present, thyroid hormone upregulates beta receptors. Epinephrine has more places to bind, and epinephrine does a better job. So we would say that thyroid hormone is permissive for catecholamines to break down fat, a process you learned from Dr. Olson is called lipolysis. We have a basic pattern of interaction, and we can talk about 3 tiers. So the hypothalamus is the first tier. It produces something called a releasing hormone. That releasing hormone is going to go through a portal system and very rapidly make its way to its target, which is the anterior pituitary. the anterior pituitary makes a stimulating hormone, which goes into the venous blood, goes to the right heart, through the lungs, to the left heart, and then out to the systemic circulation where it will find its target. In this case, our target is a peripheral gland, in this case the thyroid gland, and the stimulating hormone causes the peripheral gland to produce the hormones that we think of as having a physiologic effect. These hormones feed back and inhibit both at the anterior pituitary and at the hypothalamus. This makes sure that when you stimulate the system doesn't careen out of control. That if thyroid hormone in this case gets to be too high, we have a way of turning down the system. So the actions that thyroid hormone does, which is mainly regulating your metabolism, then metabolism is under homeostatic control because of the negative feedback regulation of these hormones. So there are many tiers or many examples of these three tiers. So gonadotropin releasing hormone sends its hormone through the portal system. The gonadotrophs release FSH and LH. They go to the gonads, and then the gonads produce sex hormone. Growth hormone releasing hormone is released from the hypothalamus, goes through a special portal system. Anterior pituitary releases growth hormone, and growth hormone causes metabolic effects. It's also going to go to various tissues, and we're going to get a hormone called IGF. IGF is a mitogen. It's going to cause cells to divide, and certainly cell division and cell growth is an aspect of growth. Growth hormone is a little more complicated because it has an inhibitory hormone. Some people call this growth hormone inhibiting hormone. It's also called somatostatin. This goes into the blood and inhibits growth hormone. So again, we want to be able to shut the system off so that growth is not uncontrolled. Somatostatin has an effect on thyroid stimulating hormone, but here is the hormone from the hypothalamus. Thyroid releasing hormone goes through the portal system, talks to the anterior pituitary, which produces thyroid stimulating hormone. That goes to the thyroid gland, and the thyroid gland produces two lipid-soluble hormones, T3 and T4. I'll show you a picture of these hormones, but these are dimers of tyrosine that have been iodinated. Thyroid releasing hormone has some influence on prolactin. So everybody has some basal level of prolactin. It has some nice effects on your immune system. But the only people that need to be making high levels of prolactin are breastfeeding mothers. So with breastfeeding, dopamine is inhibited. That releases the inhibition on prolactin, and prolactin then can go to the breast and cause milk production. Finally, we have corticotropin releasing hormone going through the portal system, activating the anterior pituitary to produce ACTH. Then that's going to go to the adrenal cortex, the outer portion of the gland, and then we get cortisol secretion. Cortisol is released with stress, so when the stress is over, we want to have a way to feed back and turn this off. So feedback is a very, very important part of endocrine control. Here's another diagram showing you that negative feedback. So we have a anterior pituitary producing a stimulating hormone. We get cortisol from the adrenal glands that's going to feed back and inhibit. Or a stimulating hormone that gets us T3 and T4, which will feed back and inhibit. We're going to stimulate the gonads. Those sex hormones feed back and inhibit. Here's some pictures from Vander showing you some anatomical considerations. Notice that the anterior and posterior pituitary sit in a bony cavity. The name of it is escaping me right now. I want to say it's the Celica turcica, and maybe someone in class can correct me, there are some neurons that are very short. So they send their product to a capillary bed. That product goes through a portal system to the anterior pituitary, and then the anterior pituitary is going to release hormones into the venous system. There are some longer hormones or longer neurons that release their hormones. That axon is going to project all the way through the pituitary stalk, synapse on a capillary bed in the posterior pituitary, and recall this is ADH and oxytocin that are released in this manner. So here's another picture from Tortora showing you these long axons projecting all the way down to the posterior pituitary through that stalk. And then we have a picture from Vander showing us a hypothalamic neuron. This is the releasing hormone going through that portal system directly to the anterior pituitary, and then this is the stimulating hormone going into the venous blood. So these are some basics of endocrinology that I hope that you've had in your previous course, that I hope you were able to review with the CT I sent home with you. And I would like to not have to lecture on this material. By all means, post your questions on the forum or email your questions, and I will certainly entertain those. And I'll end it here, and I look forward to seeing you in class.